And we will begin. Thank you everyone for coming. Your attendance is highly appreciated. And of course, if you're not able to attend live, we will be able to make the slides and webinar available after the event. So without further ado, let's get started. We're here today to talk about species-agnostic antibody discovery. This is, of course, of keen interest, I'm sure, to some of you, and hopefully we can provide a compelling case as to why. Firstly, though, I'd like to kind of go over a bit of an agenda. So we'll be talking a little bit about who we are, Rapid Novor, a little bit more about species-specific antibody discovery, as well as some case studies and some future directions for our team. For those of you who are unacquainted with us, Rapid Novor. We've been in the business of proteomics software development for quite some time, the co-founders have, but it was really about 6 years ago that they decided to focus specifically on the application of sequencing proteins. And so in that time, we've been able to help well over 400 different clients worldwide, many of the major top pharmaceutical companies by revenue, and we've been able to successfully sequence well over 3,000 monoclonal antibodies and proteins using our flagship sequencing service known as REMAB. Of course, today we're going to be talking a lot more about REPAB, and it's safe to say that we've been working on some other technologies in the past couple of years as well. Of particular note, Novor, which is useful to decoding the immunity response. And I'll be touching on that a bit further later on. And MatchMap for confirming sequence similarities. If you have protein A and protein B, of course, we can use the service to compare those two antibodies or proteins and let you know how similar they are or not, as the case may be. But without a proper introduction, I think that some of the content might be lost. So we wanted to kind of start from the ground up. For those of you who are familiar with us, sorry, you may have heard the story before, but for those of you who are new, I thought that this might be of keen interest, right? So what is de novo protein sequencing? Well, we are starting with a antibody protein. We are digesting that antibody into multiple peptides using many different enzymes. And in this way, we can cleave that antibody protein at many different regions such that the peptides will overlap over each other as we fly them over the mass spec. And in this way, we can generate many different overlapping peptide sequences prospectively from the mass spectrometry data, and then assemble them into a full-length contiguous antibody or protein sequence. So we have developed this technique, like I said, over the past 6 years, and it's yielded many completely 100% accurate proteins from our platform. And in this way, we really view it as a way in which we're turning the central dogma of molecular biology on its head. What do we mean by that? Well, of course, the dogma states that you start off with DNA, which is transcribed into RNA, which is then translated into a protein of interest. The dogma states this is the way it happens in nature, and this is the only way, the only order of operations that it follows in nature. But this is where my argument comes in, where I say, we really feel that Rapid Novor is also a part of nature. And in fact, we start with the protein of interest, we sequence it, which allows us to then recombinantly express DNA and then go through this process in reverse. And in this way, we feel like we're flipping this dogma on its head. And further, we feel that this is really a way in which we're evolving the antibody discovery process. I think antibody discovery has been a fantastic, technology. Obviously, yielded many different therapeutic and diagnostic applications, and people have benefited immensely from this technology. From the days of original hybridome development with Caesar and Colstein, but I think that much of the industry revolves around this one species here, the mice. And obviously, other species have become more well-known and studied in the past several decades. Many conventional polyclonal antibodies rely on different animal species, but I think that it's safe to say that monoclonal antibody development is been challenging in other species beyond just the mice. Of course, we've seen some in rabbits and llamas as well. But like I said, I think that really what we're getting at here is that Rapid Novor's ability to deconvolute the peptide and then subsequent protein sequence of proteins is really species agnostic. We've successfully executed cases in rabbits and canines and humans and goats. And we're further studying these other species as well and generating monoclonal antibodies directly from the serum of these different animals. So why might that be of interest? Why might we want a species-specific antibody response? Well, we like to look at canines as a good example of this. Why? Well, they're becoming quite popular in terms of their therapeutic applications and points of interest. As recently as 2016-17, approval of a therapeutic monoclonal antibody for canines from Zoetis, we see that there's a growing interest in using such technologies for our companion animals. And of course, using a species-specific antibody response can enable investigations into novel species as well. Perhaps some different animals have a more faithful representation of a human condition, as we've seen in COVID with the use of hamsters, and in that way we've benefited from that animal model. And in this way, we also can leverage a species-specific antibody response to reduce off-target binding with native mAbs, such as what we've observed with like this HAMA response in murine-derived antibodies in human patients, as I'll point in a moment. And indeed, these species-specific antibodies can also improve the efficacy and fidelity of PK assays, and further, they can allow us to take advantage of unique biologies in these non-rodent species, which are extremely well studied. But I wanted to further illustrate the point of reducing this off-target binding with these native mAbs. So, of course, there's no surprise, like this HAMA response, this human anti-murine antibody response, has been well documented in the literature for a few decades now, right? After the use of one of the first monoclonal antibodies in patients, it was observed that patients themselves actually had a background polyclonal antibody response that was binding to and neutralizing this drug of interest on the FC region. And as such, we decided to take a look at humanization efforts. So what does that look like? So we have this human of interest who received the fully murine antibody. They generated this polyclonal antibody response, which, like I said, subsequently bound to the FC region of that drug. And in order to combat this issue, we've decided to humanize these antibodies, wherein we're replacing the FC and framework regions of that original murine antibody and supplanting them with human derived constructs. And as such, we have a humanized monoclonal antibody, which is no longer dealing with this issue of FC binding polyclonal antibodies within the patient, and further reducing other problematic issues like immunogenicity. Of course, this same concept is extended to canines, and we can then caninize these monoclonal antibodies originally found in murine sources and mitigate these same binding effects that are present in those therapeutics, or even in a diagnostic context as well. And so how do we really get these naturally derived or even canonized monoclonal antibodies? Of course, there's many ways we can perform antibody discovery, right? I think the most conventional and straightforward line of thinking is to go for a hybridoma. Of course, this is where we immunize the mouse of interest with a target antigen, and we can then extract the splenocytes, perform a fusion step with the myeloma cell line, generate hybridomas, and then yield our monoclonal antibody candidates. This is a extremely fruitful, extremely robust technology that's been demonstrated over the past several decades to be efficacious in generating monoclonal antibodies, and I wouldn't contest that. What I would say is that it certainly has not translated all the way through to the canine context, right? Like there isn't a straightforward protocol for immunizing a canine, extracting some prospective B cells, and then performing a fusion with a myeloma cell line in order to generate some kind of canine hybridoma. That canonization then has to occur from these murine-derived monoclonal antibodies. And while that's obviously a good technique, it can be compared against what we're presenting further. Another option has conventionally been to use display technologies, right? Wherein we can take these B cells, we can put them into a phage or yeast display context, and we can generate a library of monoclonal antibodies against our target of interest. Again, though, we haven't seen a lot of this in the canine context. I've personally read some studies that showed, some academic labs that have done this, but to say that it is a robust and industry-ready technology, I think is a bit premature. And so as such, we haven't seen a lot of use of these kinds of technologies in the generation of monoclonal antibodies in these kinds of other veterinary or animal health contexts. One other area that we've seen where we're gaining some traction has been in single B-cell sequencing, wherein we're isolating either PBMCs or some other B-cell rich tissue, doing some kind of fax sorting step, and then we're able to perform next generation sequencing against those monoclonal antibodies, B cells, and then generate sequences which can then recapitulate the original monoclonal antibodies found within them. And again, we've seen some interest in this, I think, from the industry side, although again, I would say that this is not a robust and readily available technology. You can't just buy canine primer sets, and developing them is actually, in some cases, quite cost prohibitive. And so there's a lot of upfront investment in research and development that would have to occur prior to leveraging this kind of antibody discovery approach, right? And yet, here we are finding ourselves in the same antibody discovery approach, wherein we can just take the serum of interest from your immunized animal or animal who's a patient, and then we can perform the above mentioned de novo sequencing workflow and generate some prospective libraries of sequences. In our case, we're able to not only sequence that most abundant heavy and light chains, but we can actually, and often pair those cognate heavy and light chains together as well using our proprietary proteomics platform. So in this way, we see that there's a lot of different ways in which we can perform this antibody discovery workflow in order to generate these fully naturally derived canine antibodies. But then the question becomes, is that the only way that we can do this? So I think that extending on the first point about hybridoma development, we've seen that it doesn't typically end at this murine antibody discovery phase, right? As I mentioned earlier, there's a lot of interest in canonizing these antibodies, and that's definitely a process that has occurred readily in the industry, wherein we take these prospective murine monoclonal antibodies, we could then engineer those murine CDRs into framework and FC regions of the canine derived antibodies. And in this way, we can canonize these antibodies and allow them to be fit for purpose and reducing other kinds of off-target binding effects and immunogenicity that come as a result. And while this is a highly efficacious platform, I guess we're really just trying to demonstrate that there are other methods available as well, like certainly the approach that we've just outlined a minute ago with the serum proteomics approach. So I think that fundamentally we're getting at the difference between purely a monoclonal antibody generation strategy and a one that takes advantage of polyclonal antibodies from our target of interest. I think that really this comes down to a calculus of time and money, as they typically do, right? I wouldn't say that one is necessarily better than the other, but what I would say is that they're both complementary techniques. You're going to get something different out of the monoclonal antibody and canonization route than you would out of the next generation protein sequencing approach that we've developed here at Rapid Novor. And if you have the time and money to run them all, of course you should. And if you don't, perhaps there's some kind of calculus that is more context specific that may be of interest. I think one other advantage to point out is that we work with the native protein itself. And in this way, we can test that protein against your target of interest in your assays of interest using just like the final product. And we're also sequencing that final product. So we can observe some post-translational modifications like glycosylation on these polyclonal antibodies or the monoclonal antibodies found within the polyclonal mixture. And this way we can offer an additional piece of evidence which may sometimes escape the genetic sequencing route that you would find from the hybridoma generation strategy outlined here. So of course, this leads us to our next question of perhaps why choose these polyclonal antibodies compared to this monoclonal antibody generation strategy? And really, this comes down to a direct comparison of these PABs and MABs themselves, right? So in the polyclonal antibody case, we see that we have this multitude of monoclonal antibodies present. And by definition, they are a heterogeneous mixture of monoclonal antibodies. When we pair them with the antigen of interest, we can see that the different monoclonal antibodies can bind against many different areas of that target antigen, and as such confer binding against not one strictly defined area. Whereas, and as such, this confers some advantages. Firstly, they are easy to produce without the necessity to go through a fusion step and isolate these individual B cells. We're just immunizing and then extracting the serum of interest and purifying with the antigen of interest. We find that they're a little bit easier to produce as compared to the monoclonal antibody platforms. Of course, there's many existing polyclonal antibodies already on the market. They indeed have high affinity because of the binding of a multitude of monoclonal antibodies against their target of interest. You have the broad biophysical diversity within a polyclonal mixture present, and as such can confer greater affinities. And because of the broad biophysical diversity as well, we find that they're generally seen to be more robust and more stable in a multitude of applications as compared to the monoclonal antibody case. And of course, they have drawbacks. Namely, we have batch-to-batch variability, which is much higher than a monoclonal antibody situation because of this diversity and because of limited supply and because of challenges in upscaling that production. It's not a strictly defined singular recombinant antibody. It's a multitude of antibodies that were generated as an immune response against a target of interest. And for that reason, they also can be quite difficult to characterize because that binding is not strictly defined against like one epitope, let's say. Whereas in the monoclonal antibody case, we see that it is a homogeneous mixture of clonal antibodies by definition. And when we combine it with the antigen of interest, we see that they bind against only one region on that target. And in this way, they confer some advantages, such as recombinant antibody production is possible, resulting in indefinite access to the exact same antibody. There's obviously a lot of industry established protocols which have standardized the manufacturing process. And naturally, because it's just one antibody in this mixture, you have less barriers to characterizing that individual monoclonal antibody. And so they also suffer from some drawbacks, as I mentioned earlier, though. There's a lot of steps that are required to produce these antibodies, and they can be challenging against some targets. As I was mentioning earlier with the comparison of canonization, canonization is an art more than a science, as some would say. You start off with maybe 20 or 30 hits that have come from a murine hybridoma program, but then getting those antibodies to be fully canonized can be a challenge. It can be challenging to not break that molecule, right? Splicing those CDRs onto a canine backbone is a non-trivial task and may yield only a couple of clones at the end of the day. And certainly that can be a limiting step in this kind of approach. So coming back to this point, we feel that this is really a way where we can evolve the antibody discovery process, because we don't have to rely on these murine antibodies so much, and we don't have the upfront investment required to develop a lot of these different techniques specific for a particular species. In our hands and to us, we just see peptides, and then we can assemble those peptides into their full-length protein sequence using our technique. So with that in mind, we find that it's a nice time to launch a poll. So in your hands, what techniques are you leveraging for your antibody development? I'll leave the poll open for about a minute or two, and then we'll close and move on to the talk. Answers are coming in. Couple more seconds here. And time. Let's share these results. I should share them here. Looks like quite a few people are actually using hybridoma and display libraries for this end. And indeed, quite a lot of people are actually still using these polyclonal antibodies as well. Very good to know. So we can leave it at that and continue on with the conversation. Not a lot of single cell sequencing, I'm noticing. Yeah. And perhaps for some of the reasons that we mentioned earlier. Okay, moving on. Quickly coming back to this point about the polyclonal antibodies and some of their drawbacks, we've seen that certainly it can be a challenge to generate a highly reproducible antibody batch, right? Like you're dealing with often a pooled animal sample. You might have a roster of animals which are specifically designated for a particular project, but certainly every time you go back and create a new batch, you do have to revalidate some of those polyclonal products through a suite of assays. It can be easier if you keep the same roster of animals, but certainly it is challenging nonetheless as compared to a monoclonal antibody program. Of particular note, if you're coming to us from the European Union, we've seen that some regulators have started to consider the use of animals in any development program a lot more than they have in the past. And so this can be another a drawback of the polyclonal antibody case, as you do have to have many different live animals present to develop those polyclonals on a consistent basis year over year. And certainly, life has an expiry date for us all. And so at the end of the day, many years down the line, an antibody program may need to be significantly redeveloped and recharacterized as a result of the use of polyclonal antibodies. And this also ties into the next point of limited supply. Like I said, you only have so much. And again, there is a difficulty in characterizing this antibody response. It's not strictly defined against one particular area on a target protein of interest. And lastly, you might have the wrong species, right? For a particular context. You're not going to trick people into thinking that this mouse antibody is fully canine. Obviously, the results will speak for themselves as well. So how do we address these issues? Well, I think that it's fair. It's easier to have a nuanced conversation about one particular point of interest. And in our case, I thought it might be of interest to look at the anti-drug antibody response. So an ADA assay is of common use in the industry in characterizing the immunogenicity in response to a drug of interest. But let's quickly kind of define what we're talking about when we're talking about these assays. So we're talking about the mesoscale discovery platform, wherein we're immobilizing this drug of interest on a solid phase. And then we can flow over a monoclonal antibody, which will have one of its Fab arms binding against the immobilized drug of interest, and then the other arm binding against a labeled variant of that drug of interest. This is a nice scenario. It's just one monoclonal antibody, and it's binding against a drug with one arm and the labeled drug with the other. Very nice, picture perfect scenario, right? But of course, real life is not so easy. You often, when you're dealing with patients, have a polyclonal antibody response, which is going to bind at many different regions against that drug of interest. And this could lead to complications in interpreting that analysis with the absence of a positive control antibody to compare it against. And that's often what we have to do, right? We have to generate these blocking antibodies or different ADA antibodies, which can suffice in demonstrating the binding activity of an immune response against that drug, that therapeutic drug of interest. And so use of a well-characterized controlled reagent can kind of address these issues, right? Like if we know exactly where this monoclonal antibody is binding, what its isotype is, what its affinity is, we can really have a comprehensive understanding of what these antibodies can do when they're being taken in their context of interest. So a nice case to kind of illustrate this example, we found has been factor VIII. So factor VIII is an essential blood clotting protein for hemophilia. And we found that it was isolated from plasma originally in the early 1960s. Genentech also has become quite well known for this. They made their first recombinant in the mid 80s and then further approved by the FDA in the 90s. And this was essential in addressing this illness worldwide. However, with recombinant factor VIII, you still have this problem of immunogenicity. In fact, immunogenicity develops in 25 to 30% of all patients in the form of an anti-drug antibody response. As a result, some of these patients will be switched onto a porcine version of the recombinant factor VIII drug. And so we can see that the ADA response is actually, in effect, guiding the therapeutic the treatment of these patients with hemophilia. And so developing a robust and highly detailed anti-drug antibody assay is of critical importance to this patient base. More specifically, there have been some groups who have decided to develop a broad panel of monoclonal antibodies that can recapitulate the true immune response of these patients in response to taking factor VIII. And so there has been an effort to characterize these anti-drug antibody responses, not only in terms of their affinity, but also in terms of their binding regions. We found that studies have shown that A2 and C2 domains, two of these 5 discrete domains on the factor VIII protein, can actually lead to neutralization. And so these neutralizing antibodies are of keen interest, naturally, because they would inhibit the activity of the factor VIII drug. And further, the C1 domain has been of particular interest as well, because although it is a target for this anti-drug antibody response, it is non-neutralizing. It can lead to other pathogenicities surrounding the inability to metabolize the drug of interest, and that could be just as deleterious. And certainly the isotype can be of interest as well. Is this anti-drug antibody response dominated by IgMs? Is it dominated by IgGs? Are there IgAs present? And is there IgEs present? These can all be of particular interest in demonstrating the immunogenicity and certainly in guiding the therapeutic practice surrounding the use of this drug. And in fact, this study that I'm showing you here has shown that with patients, the factor VIII IgG4 ADA response was actually done exclusively in inhibitor patients. So in this way, we actually find that really defining the correlates can come down to the isotype and certainly the region in which these antibodies are binding. And so this makes a compelling case to answer the question of why do we need reference panels? Well, the usage of the assay can also vary greatly with no standardized positive controls. Studies have shown that in the European Union, specifically with the use of these ADA assays for factor VIII, there was between 40 and 60% variance across 15 different labs. And so naturally, the use of a standardized kit can really guide these clinical practices and create a more robust understanding of this anti-drug antibody response. And so the NIBSC and the WHO have advocated for this platform because demonstrating the efficacy using a standardized control panel of monoclonal antibodies can eliminate the issues of variance that we've observed, because some people use SPR, some people use BLI, some people use ELISA or flow cytometry. You know, this different use of these different assays can really skew these results and make them difficult to interpret on a more broad scale. And further, it wasn't just for factor VIII. We've also seen this kind of panel developed against this case of EPO. So Amgen worked with the NIBSC and the WHO to do this. They generated a panel of 9 different monoclonal antibodies targeting different epitopes with different isotypes and different affinities to offer a range of monoclonal antibodies which can faithfully recapitulate the binding activity of this ADA response in these patients of interest. So coming back to factor VIII, we see that this approach can be really useful in deconvoluting the antibody response, which we may be keen to explore further. So how would we go about this? Well, in our case, we start off with a patient or animal of interest, and then we can take our target protein and then deconvolute the discrete domains that we're targeting with our antibody enrichment strategies, such that we can generate positive selection control reagents wherein we're isolating one of these domains of interest, whether it be A2, C1, C2, and then pairing them to a FC fusion construct. And in this way, we can actually generate these positive selection reagents where we can immobilize them on an affinity column, flow through our polyclonal antibody response, and out comes the polyclonal antibody response minus the domain-specific antibody response, which we can then elute afterwards and use for our polyclonal antibody sequencing workflow, which looks something like this. So if we're starting from the bottom, because we're talking about these polyclonal antibody cases, we take these purified polyclonal antibody bleeds, we then run those purified fractionated samples through our LCM-SMS workflow. And in this way, we generate a report with the prospective heavy and light chain sequences and the paired constructs as well. And then we can work with expression partners to co-transfect those heavy and light chains and generate monoclonal antibodies, which can then be used to be studied and used in their given application, hopefully in this way of generating faithful monoclonal antibody assays. So coming back to the original case, we see that generating these domain-specific antibody responses can offer us these really efficacious and high fidelity positive control reagents and recapitulating the binding activity of these anti-drug antibody responses. So as such, let's conclude this section by saying that we have some opinions about this. From our perspective, this would also lead to the most faithful representation of the patient ADA response. You're not relying on like one antibody to confer this entire antibody response. There is in fact a panel of antibodies which are derived from this immunized patient, and it's as such, it is a more faithful representation of that immune response. This improves accessibility as well. If everyone's using the same panel, if that panel is highly accessible, this allows the use of that drug much more broadly throughout the world, because people can have a higher level of safety and confidence in using that assay because they can strictly characterize the ADA response generated after taking the drug. We're dealing with the protein, not the genes, and as such, we can feel that this is actually offering a further level of insight, such as accessing the glycosylation area found on these protein molecules. And further, we can track the parameters outside of just the binding affinities, and we can look at the anti-drug antibody itself and see if it's causing problems as well. Is this drug going to be immunogenic? Is the antibody response immunogenic? Is it aggregating and causing further problems on other tissues as well? And we may have some words to say on that a little bit later on in the talk as well. So let's stop it there for a moment, give ourselves a minute to think, and begin another poll in this section here. So let's launch. We'll give it another 30 seconds here, and then I'll close the poll. Ja, I see your question. I'll be able to answer your question at the end of the talk. Okay, let's end the poll there, share these results. It looks like most of you don't actually canonize your assay monoclonal antibodies for these purposes. There you have it. Stop sharing. Okay, this will be the home stretch here. So let's continue on. So that's the theory, folks. That's generally how we like to operate in terms of justifying these cases. But certainly, the question becomes like, how has that worked, and like, what does a project actually look like? Well, with us, we like to start off with your immunized samples of interest. If we can pull down the immunized serum and purify it against the target of interest, that is, of course, excellent material to start with. And if we can also obtain some B cells, which are from that same single source animal, that's of course of great interest to us as well. Once we have those materials, we can then screen them on our workflow. And this way we can get a very binary output, like is this antibody response sufficiently narrowed that we would be able to sequence it using our proteomics approach. And then from there, we have positive data, we move forward into deep sequencing, wherein we can use just the antigen specific serum in order to perform that sequencing, or we can complement that data further with a transcriptomic analysis of the B cells that we've identified from the bleeds. Once we have these prospective heavy and light chains together, we can pair them into the cognate full-length IgG molecule. And once we have that, we can then express the sequence recombinantly and send off these antibodies for testing in your hands. Of course, that's the idea. How did we do? So we've decided to do our first few batches of projects in rabbits. Rabbits are nice because they have excellent properties for immunohistochemistry. They work really well against small targets, particularly peptides and small molecules. They have a unique ontogeny, and so they generate some different monoclonal antibodies that you may find in other rodent species like mice. And lastly, they have a really strong, robust structure due to the interchain bonding cysteines between C80 and C171 on the light chain. And so another point was that these animals are big. Our techniques have been developing, our workflows have been evolving, and we've been constantly reducing the amount of sample that we require in order to run this given analysis. We might have some more to say about that in the coming weeks, but I would say that rabbits were nice because we could sufficiently extract serum to perform the antigen-specific purification and then run it on our workflow, which was nice. And further, B-cell repertoire sequencing primers are readily available, as a kit, and so that was also helpful in our first few experiments. And so we found that there were many different applications that were of keen interest. Of course, therapeutic antibody development, characterization of these immune responses, the diagnostic development side of things, but a lot of people just have access to polyclonal antibodies that they would like to convert into a monoclonal antibody or mAb cocktail. And that's where we started. So we immunized our rabbit with a peptide antigen, and this is some of the original data that we got. So this was from last year. So as I continue to show you some of this data, keep that in mind. This is actually data that was generated well over a year ago now. And so for those of you that are unacquainted with our sequencing workflow and data reporting, what you're seeing in front of you is the sequence alignment data for the heavy chain and the light chain. This top bar here denotes the region of the antibody. Your green is for your frameworks, your CDRs are in orange, and as we go to the right, we find that eventually we see the FC region, the constant region. And this, of course, extends further, but we're not going to look at all that. We're looking at just the variable region in this picture here. And so as you see, there's different peptides that are generated below. Each one of these individual lines is an individual peptide read, and the different colors denote different enzymes that were used in the sample prep for the sample. So we have different enzymes used like pepsin, chymotrypsin, trypsin, different enzymes confer different cleavage region spots. And so for that reason, we like to have many different enzymes that can digest these antibodies at many different spots, and then they can have overlapping peptides all the way across. Okay, so with that in mind, what's a good data look like? Well, in our perspective, we like to achieve at least 20x coverage all the way across the entire sequence of the heavy and light chain. And so that means that we have at least 20 overlapping peptides over every single amino acid. In this case, that threshold was met. The heavy and light chains looked quite good all the way through. We felt happy in expressing these constructs. And further, this other antibody in this original case, antibody 3, looked quite well for similar reasons. And so we then expressed and tested these monoclonal antibodies from the polyclonal serum by ELISA, and indeed they perform really well. The original polyclonal mixture is shown here in blue at PD030. And as you can see, R1 and R3 faithfully recapitulate the binding activity of that original polyclonal antibody. And so we were quite happy with these results. We further demonstrated the efficacy by SPR, and we found that the original polyclonal antibody and the two recombinants all had low single-digit nanomolar binding affinities, and as such, we were happy to see that these antibodies have a very high affinity against their targets of interest. These are actually internal reagent antibodies that we use, and we still use them quite well today for immunoprecipitation applications. So next, we had a client come to us and decide to work on diagnostic development. They wanted us to generate a monoclonal antibody against a peptide of interest from a biofluid of interest, so that they could then purify this particular peptide and then perform a quantitative assay and infer the level of that peptide. So they left us in a bit of a time crunch. This project needed to get done quite quickly within a couple of months. And so we were luckily quite successful in generating these highly specific, high affinity antibodies against this target of interest. And indeed, they were fit for purpose in their intended application. And so this is the ELISA that we generated from the proteomics only derived clones. This is what I meant by a bit of a time crunch. We were generating NGS data as well, but at the time that they needed the antibodies, we only had proteomics data to use. And in fact, we were able to successfully sequence 6 monoclonal antibodies from only the proteomics, so just directly from the serum. As you can see here, the original polyclonal is in red, and these other 6 antibody constructs faithfully recapitulate the binding activity by ELISA and also in their intended immunoprecipitation context. Further, when we did have access to NGS data, we were able to subsequently generate another 6 monoclonal antibody sequences, which were unique. And then in total, we generated 12 fully paired IgG constructs, which were obviously quite efficacious against their target. As you see here, they all perform quite well by ELISA, and again, in immunoprecipitation assays as well. So that was obviously quite a good case. We're happy about that, and we've moved on into exploring other novel species. So we also had the chance to work with a partner who sent us a goat polyclonal, which was immunized with a peptide. And we found that really this kind of off-the-shelf tab had 2 major forms that were amongst the polyclonal background. There were dozens of other antibodies present in that sample, but there were really 2 that stood out in significant quantities as compared to that polyclonal background. And this is some of the coverage data. We are anxiously waiting for the binding data and the rest of that data. But as you can see, we had excellent levels of coverage all throughout this monoclonal antibody sequence. And as such, we're quite confident in the binding activity that will follow upon seeing the recombinant antibody expression. And so we see here that we've been able to successfully sequence antibodies in rabbits, in goats. We've also ran some projects in canines and in humans as well. And we've really been able to, over the past year or so, been able to demonstrate this species-agnostic approach in delivering species-specific monoclonal antibodies. The last case I'll show you is actually in hematological malignancy. So we've done some work in multiple myeloma. In sequencing the monoclonal protein known as M protein in patients. I show this because, of course, it is of some interest to some people working on canine oncology as well. The ability to actually track the clonal evolution and quantity of a monoclonal protein may be of interest in diagnosing disease. We found that it has been for the patient context as well. So the problem is often that in these hematological cases that there is a particular clone, of monoclonal antibody, which is overexpressed to the point where it is actually pathogenic to the patient of interest. This can lead to kidney issues and other kinds of pathologies where the monoclonal antibodies are actually accumulating on tissues and causing problems. And so there are 2 conventional assays that are used to diagnose the condition as it is, but they both suffer, or sorry, I should say there are 2 drawbacks of conventional assays that are used to diagnose this condition. The first is that they can often lack sensitivity if you're using immunofixation or immunoprecipitation. These different assays can be just of low sensitivity, and the problem is, in the absence of higher sensitivity, you may change the treatment regimen without the knowledge that a patient's monoclonal protein is actually on the rise again. And further, there are some assays which can use a bone marrow biopsy, but these kinds of biopsies can be highly invasive for our patient population, and as such, they may opt not to do them as often, particularly if they're geriatric, which is often the case with multiple myeloma. And so Rapid Novor has been able to develop a assay to address these issues. By just performing a blood draw, we can actually put the serum extracted from these patients through our LC-MS/MS workflow and sequence the most dominant monoclonal protein found. And as such, perform a subsequent follow-up where we then pull some more blood and then can actually quantify the abundance of that monoclonal protein as it evolves over time using longitudinal samples. So this is some of the results that we've had in a recent publication. We can really demonstrate the efficacy of the platform in this way because we can demonstrate to a very high level of sensitivity that these patients are low in terms of their levels of M protein, or in fact, very high, as the case may be in the case of a relapse. I think it might be nice to kind of zoom in on one of these cases. And this patient in particular, 004-045, I thought was of particular interest. Why? Well, because as we look at this table here, we can see the status that was reported off of the conventional analyses that were performed. And we see that they were in clinical remission as of day 100 post-treatment, but they stayed in clinical remission all the way through up till month, up until month 30, but even in month 24, they were still being reported as in clinical remission and MRD positive. And so I point this out because as we see from our analysis, we using our M protein analysis can observe that this is actually looking a lot more like a relapse than it is a clinical remission. And in fact, on the month 30, we see that it was a progression of disease again. And so the sensitivity of the assay has been explored, and it looks quite promising in terms of its applications. And this has yielded a couple of papers that we released just a few weeks ago. Feel free to check them out. Of course, you can email me personally as well, and I'd be happy to talk. But of course, I show this case because I show it to demonstrate the efficacy of not only in our human-centered approach, but also prospectively in other samples from other species as well. So what do we need to really get this project started? Well, really, if you just have serum against your target of interest, we can work with that. It's quite helpful to also have a bit of blood so that we can pull out some B cells from the PBMCs, or if it is possible, splenocytes as well, but that's not strictly speaking necessary. We can just work with a blood draw, keep the animal alive, and then perform the serum proteomics to derive those monoclonal antibodies. And in terms of future developments for the company, we're always working on something. In particular, we like to highlight that we're working on custom NGS development. We've just recently begun to onboard our NGS lab. We're always improving our sequencing capabilities from the protein side. We're always looking to find more antigen-specific leads and novel methods for immunizations as well. And in this way, we're generating species-specific antibodies, which can be sequenced directly from the animal serum. And we can generate a library of antibody sequences using either the proteogenomics approach or the protein-only approach. And in this way, we're seeking royalty-free contracts for access to the technology so that we can run more projects and deliver more antibodies and help you. So without further ado, that's it. My name is Anthony, and if you can feel free to contact me either by email or just reply to one of our invitations, I'm sure we'll be able to get back to you. As you can see, this is a pre-COVID haircut. Looking forward to meeting you guys in this well, hopefully post-COVID world in the coming months and years as well. So thanks so much for attending. I'll be able to stick around a little bit longer and answer some questions. And like I said, if you don't have any immediately, feel free to reach out to us afterwards as well. We'll be happy to hear back from you. So I do have a question in here. I see that she has asked, how do you restore the original pairs if the sequence sample is polyclonal? It's a great question. Of course, everyone's always keen to know how we're doing the pairing. And what I can say is that we have the advantage of working with the native molecule. We have a number of different purification strategies that we can deploy against the polyclonal antibody mixture. And then we can use a suite of algorithms that we've developed to actually pair those antibody constructs, like I said, using the native molecule. So an intact mass approach may be of interest, but there's also a handful of other experiments that we can run in order to derive that pairing combination. And of course, I'd be happy to maybe detail that in a bit more of a private context. But of course, yeah, we can set something up by email as well. So feel free to reach out.